Today we will talk about the Campylobacter bacteria. You have to remember the two most common species of the Campylobacter. First one is the Campylobacter jejuni and the second one is the Campylobacter coli. Next, we will talk about the characteristic of the Campylobacter. Campylobacter is a gram-negative bacteria. It means that it will look pink through the gram staining. And if we talk about the shape, it is curved or a spiral shape. So that's why we called it comma-shaped bacteria. It has single polar or bipolar flagellum. So that's why it is good in motility. Oxygen requirement, it is microaerophilic, means it requires low concentrations of the oxygens like 5 to 10% oxygen. Optimal growth conditions, Campylobacter species thrive at 37 to 42 degrees Celsius, making the GIT tract of warm-blooded animals as ideal environment because the warm-blooded animals maintain their body temperature. So, it is good for the survival of the Campylobacter bacteria. 37 degree to 42 degrees Celsius. Next, if we talk about the epidemiology, global impact, Campylobacter infections are among the leading causes of the bacterial diarrhea globally, specifically in the low and the middle income countries. Source of infections like primary reservoirs, poultry, cattle, and other domesticated animals. Human acquired the infection through consumption of the undercooked or contaminated meat, specifically chicken, drinking contaminated water or unpasteurized milk, contact with the infected animals or their feces. So generally, animals are the sources of infection of this Campylobacter. Incidence, according to the WHO, an estimated 1 in 10 people worldwide suffer from the foodborne illness. Next, we will talk about the pathogenesis, adhesion and colonization. So bacteria will get chance to cause infection when bacteria attached to the surface of the body. So in that case, Campylobacter has flagella. It means that it can move easily in our GI tract. And Campylobacter also have adhesins that enables the Campylobacter to attach to the surface of gut. Invasion, Campylobacter invades the epithelial cell leading to localized inflammation. It means that they can invade the epithelial cells of our GIT tract and cause the inflammation at that site. Also, most important, it produces the toxins such as cytolether distending toxin CDT, contributes to cellular damage and the diarrhea. Next, we will look at the manifestations. Campylobacter infections, also known as campylobacteriosis, most commonly GIT symptoms like diarrhea, often bloody diarrhea, abdominal cramps, fever, nausea, and vomiting. That is the most important one. Complicated things or complicated manifestations like Guillain-Barré syndrome, it's a rare one, but you will see post-infectious complication and cause the flaccid paralysis in the Guillain-Barré syndrome. Reactive arthritis, inflammatory joint conditions may be developed uh, in that case. Bacteremia occurs rarely, but particularly in immunocompromised patients, like bacteria can enter in our bloodstream that cause the bacteremia. Next, how we will diagnose the Campylobacter? First one is the culture. Stool samples are cultured on selective media under microaerophilic conditions, as we have discussed. This is the favorable conditions for the Campylobacter. Next, molecular methods like PCR, we can detect the genetic materials of the Campylobacter species through the PCR polymerase chain reactions. Microscopic direct examination of the stool samples using phase contrast or dark field microscopy reveal the spiral shape microorganisms. If we talk about the treatment, first one is the sportive care. Rehydration and electrolyte replacement are the mainstay of the treatment of the patients because during the diarrhea, a lot of water waste from our body. So in that case, rehydration and the electrolytes must be given to the patient. Next is the antibiotic. Severe cases or in immunocompromised uh, patients, you will see the macrolides, azithromycin, and the fluoroquinolone, ciprofloxacin. And next is the antimicrobial resistance. The increasing resistance of Campylobacter to the fluoroquinolones is growing public health concern. And most probably, and it is good to decide which antibiotic is given to the patient after the culture. So culture will decide the best antibiotic given to the patient. Next, prevention. So, food safety practices, proper cooking of poultry and meat, avoiding cross contamination in the kitchen, consuming pasteurized milk and treated water. Personal hygienes like hand washing, handling raw meat, interacting with animals, or using restroom. Next, public health measures. Surveillance and regulation of the food production processes to minimize contaminations. So this is all about the Campylobacter. If you still have any question, you may ask in the comment section. Thank you so much.